Don't we? Okay, I think we should probably start now. It's 4:02. So, yeah, let's just formally get started. Robert, is that okay? We can start.
I think we're all set up, yeah?
Okay, great. So, do you want to do the intro?
Go ahead. I'll let you
Oh,
lead
okay.
us.
Okay, good. Okay, hi everyone. This is the Longevity Biotech Show with Nathan Chang and Robert Zimin. Today we have the great privilege of having Matt Scholz, the co-founder of Oisín Biotechnologies, here with us. Oisín Biotechnologies is a gene therapy company that is targeting senescent cells. So today we're just going to talk about Matt's backstory, how he got into longevity, days of the company, how he built it out, and maybe this can help serve as just a reference or a template for other people who are thinking about getting into longevity, maybe starting their own company. So without further ado, let's just start off. Oh, actually, housekeeping. This is being recorded, so later on, when we open up for questions, so after maybe 30 minutes, just FYI, if you come up to ask a question, that means you're consenting to us allowing us to use your voice in the recording, the podcast, and also your profile photo when we upload the recording to YouTube. Okay, so let's just start with some quick introductions. I'll go first, and then Robert, and then Matt. So myself, I'm Nathan. I'm the founder of Longevity Market Cap and the editor of the Longevity Market Cap newsletter. It's a once a week sort of roundup of things that are going on in the longevity biotech industry. I'm also the founder of Longevity List, which is a website where you can find jobs, companies, and investors in longevity biotech. Yeah, so Robert, would you like to introduce yourself?
Thanks, Nathan. Yeah, so I'm Robert Zimin. I am a researcher currently affiliated with the Cohen Lab based outside of Montreal in Quebec, Canada. I have about 10 years of experience in bioinformatics and research software engineering and have been getting more involved in the longevity biotech space in the last year or so. Matt?
I'm Matt Scholz. I'm the CEO of Oisín Biotechnologies, and it's an oncology-focused spin-out, Oncocenix. And I guess I'll save the rest for probably as we go through this. Don't need to do the bio in advance.
Okay, great. Sounds good. So I guess we'll start off by focusing on the present, and then after we cover that, so that we have some context about Oisín Biotechnologies, then maybe we'll sort of rewind and start from your beginning. Yeah, so what is Oisín Biotechnologies?
So we're a company focused on basically treating diseases of aging with the goal of extending health span and eventually lifespan. And we kind of got our start as a new class of senolytic that could basically kill cells based on what they were thinking, as we like to say, but based on their transcriptional activity. So instead of like using a poison to like a chemotherapy to kill these things or trying to find an antibody against them, we can effectively upload a program into your veins that says, if you're senescent, kill yourself. And but we're really looking at senescence as kind of the tip of the spear, and our goal is to go after a lot of other things. And so we have this platform that allows us to produce proteins kind of arbitrarily in different cells in the body or just all over the place. And so we're kind of setting out at a senolytic because it's what we can address today. I think that people have kind of accepted it as a viable target and believe there's benefits in doing it, and we can tie it to diseases the FDA views as druggable targets, and then go from there.
Great, yeah. So your technology right now, the gene therapy targeting senescence, can you just give us a little bit of background on like what the target is specifically and how you're delivering this gene therapy to these senescent cells?
Yeah, so the first target we had selected was P16, and we did that largely because of these studies out of the Mayo Clinic where they showed the benefits of ablating P16 positive senescent cells. As as the field grows, though, I'd say maybe take a step back. I don't think the field has yet to characterize every population of senescent cells in humans. I think there are kinds of senescent cells we don't yet understand, and some of which we don't even know exist. But I'd say P16 is probably the most validated, broadest, and most specific at the same time as a marker for senescence that you can actually get at. And so we set out to simply build a clinically viable version of what was done in the Mayo Clinic studies. And for those who aren't familiar with it, the Mayo Clinic and the Buck basically built some transgenic mice that allowed you to kill P16 positive cells with an otherwise innocuous drug. And they use this to kill those cells intermittently, in some cases starting after weaning, and in some cases in more middle age. And they showed this like really compelling data, like that if you started at a relatively young age in particular, most of the pathologies of age didn't really manifest themselves at all. They were healthy, they're less likely to get cancer, and they lived longer and then died abruptly, basically. And the real problem with that, I suppose, like a lot of academic studies, is that it's not even remotely translatable. Like in order to do this, you have to go back in time and modify your own embryo, the GMO mouse. And so we set out to do that in a way that would be clinically viable, but still using the same kind of thesis. So if you consider, like, say, the dasatinib and quercetin treatments, these things are, one's a chemotherapy, the other's a flavonoid. Like they're killing a cell effectively based on metabolic attributes, and it definitely skews harder to P53 senescent cells than P16. And so we wanted the straight P16 one initially, and that's what we built and what we generated a lot of our data with. But the beauty of this approach is that you can target multiple pathways at the same time. You can basically put Boolean logic into DNA. So I'd say the most effective version we've got actually combines P16 and P53 targeting. And so we get complete knockdown of both in a construct that you can deliver across the body without having off-target effects. And so as we go forward, I think kind of the holy grail in the field is to build a true pan-senescence promoter, something that works in all the different types of senescent cells and not the bad. And I think that to do that in practice will require combinations of promoters and repressors, or promoters and interfering RNA, something to focus it a little bit better. But that's where we're going with it now.
Right. So if I understand correctly, you have this gene therapy that gets activated from these promoters of markers of senescence, and then it sort of induces like an apoptosis, and the cell just commits suicide or something like that.
That's right. Yeah. So I don't know how technical the audience is, but you have so feel free to ask questions. But I like to say I'm fluent in English and gibberish, so I can be pushed in either direction pretty easily if I go too far one way. But you have a promoter that can drive pretty much any gene you want. In this case, P16, and so P16 is a tumor suppressor, is what its kind of mission in life is normally. But it's also activated in senescence, and I mean senescence can be thought of as a kind of a last-ditch attempt to avoid cancer as well, because basically when a cell becomes senescent, it's not able to divide anymore, at least not unless you mutate it. And so this thing goes active, and we know it's on in the majority of the senescent cells. So what we did was we put a suicide gene, an inducible version of caspase-9, under that promoter. So if P16 goes up, suicide gene gets made, cell gets killed. If P16 is not high, nothing happens, and the cell goes about its business.
Okay, awesome. And how do you deliver this gene?
That's a great question. So, I guess if you think about gene therapy, like people have wanted to mess with DNA ever since they knew what DNA was. But the Achilles heel of this field really has been delivery. Like, how do you get your genetic payload into cells across the body? And, recognizing that your body has like a vested interest in not allowing this to happen. I mean, it basically thinks virus when it thinks foreign DNA. And so your body has all these defenses that would, if you just put DNA into the blood, it will be enzymatically degraded. It doesn't enter cells efficiently. And if you use a virus, you have even worse problems in that respect. But it protects it in the blood, but now your body knows it's a virus. So what we use is something called a proteolipid vehicle, and this is a subset of lipid nanoparticle. So think of like microscopic fat bubbles. You know, they're like 80 nanometers across, really, really small little fat bubbles with DNA on the inside. And if you've been following that field, you've probably seen that they made a bit of a Faustian bargain of sorts. And that if you take a neutrally charged lipid, it's well tolerated. It doesn't hurt anyone if you inject even large amounts of it. But it also isn't very good at getting into cells. They don't work very well. And so if you look at like the formulations used by companies like Moderna, these are charged lipids and or conditionally charged lipids, and basically they have a positive or negative charge or one that can be induced, and that allows them to get into cells relatively efficiently, but it also leads to them being wildly toxic. So if you inject these things IV, you can only put tiny amounts in before you destroy the liver, basically. And a good example of this is the COVID vaccine for Pfizer and Moderna. Like they're administering millions of a gram of these things to full-grown men, and so it's a tiny amount, and you still will get some toxicity from it. And so they couldn't go any higher in the vaccine without causing toxicity that was too bad for it to progress. So I mean, they've been working on these things for a long time. And so the proteolipid vehicle is neutral, and the one we use, so it doesn't have any of those issues. But to get into the cell, we have a tiny little viral protein on the surface, a fusogenic peptide. And it is quite minuscule. Like the part that sticks out is only 12 amino acids long. So it's like 1,000 times smaller than, say, like the spike on influenza or something that would do something similar. And that little peptide is able to mix the lipids of the particle with the lipids of a cell membrane. And so it basically goes right through the membrane and delivers the payload to the cytoplasm. And it is important because it doesn't need to be endocytosed. It doesn't rely on any kind of uptake from the cell. It just indiscriminately fuses with everything. And so philosophically, this is actually a pretty radical departure. Like what we've opted to do is take targeting out of the realm of chemistry and bring it into the realm of information. So we have an utterly indiscriminate delivery system with a highly targeted payload.
Right. So the idea is, I guess, to just get the delivery vehicle into all cells and then let the gene do the targeting.
That's exactly right. I mean, to hit
Okay.
every cell in the body is probably not practical, but we can hit lots of them. I mean, the majority of them probably. There's nothing else that comes even close to the penetration we can get. I mean, if you look at organs that take a lot of it, like we hit pretty much every cell in the tissue, and so it doesn't cross the blood-brain barrier, notably. You can administer it into the CNS and it will function just fine there, but it doesn't cross really on its own. So you don't hit like every cell in the whole body, but we hit an awful lot of them.
Okay, that's really cool. So where are you guys right now in development?
So the most okay, so on the delivery side, the most advanced thing is actually a COVID vaccine, which is in clinic already. On our side, the cancer program is a little more advanced than the aging program, and that's already had its first regulatory meetings. It basically just has GLP talks ahead of it. For the aging front, and like so, there's this overlap between aging and cancer. You can almost use the two interchangeably. Like we definitely optimize for both, but there is a lot of overlap. Like it's amazing how many cancers you could kill with something that targets these genes. And so on the senolytic side, though, our first target is kidney disease. And we're looking at it both in the ability to prevent the transition from an acute kidney injury into chronic kidney disease and the ability to like arrest progression of the chronic kidney disease. And so that basically senescent cells are known to play a role in the pathogenesis of chronic kidney disease. They kind of drive this vicious cycle of inflammation, fibrosis, more senescence, and that keeps going until you end up in end-stage renal failure. So if we can kill off those senescent cells, the idea is that the kidney can recover.
Okay, great. And do you have like a timeline for when you think you might be able to, I don't know, maybe get to the clinic?
Well,
You guys doing
a lot
IND?
of this, yeah, I mean, I'd say we're gated more on funding than we are on science, and so we're actually just wrapped up a seed round, and that will take care of the rest of our preclinical studies. Then we have the regulatory meeting for that, and we'll go into the clinic. I think we're probably around 18 months away, but assuming things go relatively smoothly in biotech, at least as smoothly as biotech goes.
Awesome. Well, that's really exciting. Okay, great. So now that we have like a bit of context about Oisín Biotechnologies and the approach, maybe we can rewind to sort of the earlier days of the company so that we can give people an idea of sort of what it takes to do something like this. So maybe you can start by just telling me how did you start the company? Like how did it actually form?
Yeah. So I guess if we go back a little further, my background is in computer science and physics, not bio. And so the perspective, I suppose, taking going into this is a little unconventional. And the North Star for me really was that the essence of life is information. And if you can manipulate the information in the body, you can do anything. And for pretty much all of human history, we've approached medicine from the perspective of like plants and prayers, small molecule drugs and ritual. And the ability to manipulate the code of life really opens up whole new doors. And so that's the perspective that led me to here. But the genesis of Ocean actually was quite interesting. It came kind of out of a company I was doing before, so Immusoft, which is my first foray into biotech, and we were building what amounted to an app store for the human body. So producing long-lived plasma cells that secreted therapeutic proteins and the ability to just upload new programs to your body to make new things you wanted. And one of the long-standing goals I had for the platform was to basically try to recreate the biochemical environment of youth as you aged. And I mean, if you think about it, if you took your blood, once a year from birth until death, and you looked at what was inside and floating around, it wouldn't be the same. I mean, obvious things change, like say testosterone or estrogen, but countless other things change too. You get accumulation of bad things, reduction of some good, and you can never take enough drugs to put it back the way it should be. But if you can program your body to make things, you can have them secreting these things 24/7 or secreting things that clean up the mess 24/7. And so that's kind of this long-standing goal. And I was giving a talk about this at a health extension salon put on by Jobette Lacroix in the Bay Area. And and I spoke right before Judy Campisi spoke. And she was actually presenting some of this early data from the collaboration between the Buck and the Mayo Clinic. And at the time, I wasn't really following the space. Like I didn't know much about senescent cells or anything like that. And I was watching this data and kind of listening to people muse about how to translate this. And I looked at this guy across the table. I was like, "That's fascinating, but I do it entirely different." And this guy is Gary Hudson, who ended up being a co-founder. And at the bar afterwards, he said, "How would you do it?" And I kind of spit out this idea of trying to kill off these cells with a genetic program. And he thought this was a great idea. And he's basically, we should start a company. We got to do it. And I just closed my Series A for Immusoft. I'm like, my investors will, castrate me if I start another company. And so I don't know what I can do about it. And he's like, "Well, I tell you what, if you basically write it up, I'll put in the first money and I'll run it until you can take over. Eventually, you'll exit Immusoft, and then I can raise more money, but it just has to happen." And so I thought this was a pretty compelling pitch, and we set to work building basically a plan, and we built it to basically fail fast. Like we had really brutal stage gates on it. I wanted it to, if it was successful, it had to be really successful, really compelling, because I didn't want this thing like, wasting time basically. And so we set out these stage gates and we licensed in the technology to do it. And the first data was basically better than we'd expected. And so it got everyone really excited. And that's kind of how it went going. He's able to bring in more money. And it was a couple of years later that I finally transitioned into being the CEO.
Okay, awesome. Yeah. I actually want to, yeah, maybe dig deeper on this these gates that you're talking about, like a de-risking process. So when you started the company, what were those sort of milestones that you wanted to hit?
Well, I mean, the big one was really, can we kill senescent cells in a living organism with this treatment? And so there's, I mean, little ones about, of course, putting the whole thing together and making sure we could, even assemble nanoparticles that did this correctly. But then, yeah, it really came down to, can we take a mouse, induce senescence, and in this case, so we give it, like, back then we didn't have enough money to naturally age mice. So we give them basically high doses of chemotherapy and radiate them and stuff, and you can see senescence accumulate if you are using a luciferase mouse for this, or you stain for it after the fact. And so we found that we could induce the senescence and then give it basically a single treatment with this nanoparticle and knock it down again. And so that was really the primary gate. Like, could you actually use this in a living organism? And I said normally, like, one could easily put together, NIH grants to explore the periphery of this for years before getting to the point of saying, I want to see if it works and doesn't, kill the poor mouse. And we pretty much went to that straight away, because I thought if we couldn't, hit those cells in a living organism out of the gate broadly enough with like a practical dose, this is going to be an even steeper uphill battle than it would normally be. And so that was our first shot.
Okay, great. Yeah, and that is an interesting point about not being able to afford the naturally aged mouse, because that's something that we've talked about with other people, other co-founders on this show. Maybe you can, I don't know if you can tell us, but just like from a practical standpoint, starting a company and getting the first data, how much money would someone like a founder today need to put in to get something like that, like the data that you were getting to first de-risk the company?
That's a great question. It depends far more on how creative you are than I think anything else. I mean, the vanilla answer is probably a few $1,000,000, but you can do it for way, way less. And so one thing I'd noticed in kind of my first journey into biotech was that there's a great deal of latent capacity in academia. Academic labs have interesting, cycles, and they, get a grant, get some data, publish, get a new grant, and frequently, like, just due to how long the grant cycles take, they'll have actually done the data or done the research before they even get their grant, and then they do things like approach the end of the grant, and if they still have money left over, they buy as much equipment and goodies as they can, because they don't want to, like, not use all the money. And so they end up with some interesting kind of artifacts of this. So that there's often more equipment, more resources sitting in an academic lab than you might expect. And so it's kind of, I guess, disproportionately allocated. So there's definitely some starving young scientists. But if you can combine this with something new and attractive technically, and often get really fruitful academic collaborations. Like, I've made some really interesting deals with academic groups where I basically, like, I have this cool technology. If you kind of test it in your lab with your stuff, then you can publish on it, and I get to just use the data. So my general philosophy is they can have the glory. I just need the IP. So, because you're trying to commercialize this, not get a paper published. And so you could, I mean, depending on what you are actually testing, you do things really cheap on a shoestring. I mean, I had, like, on my first company, like, the first, like, license was a few grand. I got, like, the lab that made it, or who I licensed it from, to, like, build me the prototype. So, and then I made another deal with a different lab so that they could use it in some of their research. If I could get, like, visiting scientist status there and test it on the cells we wanted to. So you can be very creative, and I think you really need to be, unless you are just one of the lucky ones who come out of a prominent lab and have a big VC that wants to fund it. But even if you have that as an option, I mean, there's certainly downsides to it. Like, the VC will own everything from the beginning, and you might not have the freedom you want to go in the directions you want with it.
Right. So doing this sort of like shoestring approach, what kind of order of magnitude do you think it would cost
Well, like I
to
said,
start
it
something?
depends a great deal on the kind of work you need to do. Like, in vitro work could be done almost for free, really, because if the reagents aren't particularly expensive, it's a few hours on some machines, a little bit of tissue culture time maybe, and you're good. If you need to do in vivo studies, like, that will definitely complicate things, because you have to have a vivarium that's able to, do that kind of work, and they can be harder to do for cheap or free. But I mean, I had a really fascinating project I did years ago, and it was during the Ebola outbreak, and I'd been working on this tool to basically do gene therapy in the garage. open source in vivo electroporator. And I thought this had the potential to bring expensive biologics into the developing world, where you wouldn't have a cold supply chain and wouldn't have, necessarily all the infrastructure you need to otherwise use them. And we ended up doing, it was a primate study, so what would have been a very expensive study at the university, and basically all we ended up really paying for was the DNA we put in, and which is actually a lot of DNA. It turned our lab into like Breaking Bad for a week, as we had like every container we could find was full of E. coli making DNA. But basically, these animals were coming off protocol for something else. And so they're able to do, imagine a protocol amendment to test it on this. And it basically ran through on the same budget and everything and under the same auspices. So, I mean, normally to do a primate study is like, it costs you $100,000 or something probably. And, or more depending on what you want to do with them. And in this case, it was like a few $1,000s. We went from an idea to actual data in primates, which is pretty amazing. And so you can, I said, it's really limited mostly by your own creativity. You can, if you have a motivated academic partner, you can accomplish quite a bit. And I actually think that in some respects, this is better than just having a ton of money out of the get-go, because you're kind of forced to get these guys to buy off on it. Like someone who's running a lab with these resources is not going to like allocate time, money, reagents to doing it if they don't think it's interesting and has promise. Because what's in it for them, I guess. But in the process of getting these people to buy off on it, you might also be working with them on the experimental designs and whatnot, and realize maybe shortcomings you had in the initial one, or some other things you should look at. The ad hoc interactions. Whereas if you just had a CRO do it, you write the check and they do it, and for better or worse, data comes out the other end. So the process of building it organically, actually, I think is attractive. Even now, having a lot more resources than I did before, we still do these kinds of collaborations, and basically find some people who are really bright and motivated in academia and say, hey, we'll give you a tool that nobody else has, and run these experiments and and see what happens. And it's even right now, like we're doing some collaborations that are pretty cool. Like we're seeing data that I don't think anyone really expected. It's exciting.
Yeah, that's great. Yeah, I really like that approach, and it makes sense from a practical standpoint and also a scientific standpoint. So very cool. Um, Maybe I can just ask one quick question before I hand it over to Robert. So for founders who are starting off today, who want to do something in longevity, what sort of words of advice do you have for them? Like do's and maybe some do nots?
Well, it's interesting about the space is it is still quite young. And I mean, when I first got into bio, I was trying to do something no one else was trying to do, and I thought that was both a blessing and a curse. Like in that, like my relative lack of experience didn't count against me as much as it might otherwise, because no one had done it anyway. And so it was definitely spend more time trying to figure out what didn't work previously than trying to even figure out what I wanted to do. And I think in some respects it's probably a good piece of advice. Like if you're hatching up something, it is definitely good to get as much feedback on like why it won't work as possible. And ideally, you find someone who tried it and failed, so you can understand why. And this is something that it's a little counterintuitive, but I think was very helpful early on. Is I would talk about what I wanted to do to everybody who would listen, and the feedback was generally like, "You're crazy. That won't work. You don't even have a business being in this field in the first place." And but that kind of feedback really allowed me to basically hone the strategy. Get rid of some things that would have probably been obvious problems that weren't on my radar before. And in fact, every time someone would say something like that, I would kind of pin them down and be like, "Okay, what's the number one reason this won't work or is a bad idea?" And I would go dig into that reason in particular. I'd find experts in that area. I would call them up and say, "Hey, this is what I want to do, but I think this is a problem. Can you help me solve the problem?" And until eventually there were no more glaring reasons. And then we actually got funded, and that's how we launched. But I think that if you're looking at starting a company in this field, like you have to kind of look at the landscape of what's going on now, what's been done, and decide kind of where you want to fit into this. And like general attitude is I like the ambitious projects. Like in my mind, even starting something simple like a falafel stand is hard. Like any business is hard. It's going to take your time, your money, your youth. The So do something that you're going to be really happy with when you trade all those things for it. That has a real upside. And going after aging as a whole, or gene therapy and biotechnology, these are high risk, high reward fields. And so I think really you've got to know what it is you want to master and what the outcome is, kind of before you get into it. And my, I guess, general philosophy with these things is to be the undisputed world expert in what we were doing and effectively nothing else. To basically rely on others like who are really good at those things to do those things and focus on our core technology, why it was different, why people need to do this exact thing and not something else.
Awesome. Yeah, that's really good advice. I, I think that's also interesting, the point about just asking everybody like why won't this idea work. I think in biotech a lot of people are very secretive about what they're up to, so they probably don't want to share their approach with other people. But I guess the quickest way to de-risk is to ask experts.
What is funny, like
Yeah,
a lot
so
of
oh,
I say a lot of like I think first time entrepreneurs make this mistake that like their idea is super brilliant, they're the only one who's thought of it, and if other people hear it, they're gonna steal it. And in my case, it's rarely happens. I mean, for the most part, people think my ideas are stupid, so they don't try to steal them, which is a big plus to doing something new. But even if someone wanted to, I mean, I think there's ideas are cheap, execution is hard. And so there's lots of people who hatch up ideas all the time that don't go anywhere with them. And so that definitely the more things you solve, like you begin to accumulate, intellectual property that you probably don't want to share with everybody. But I don't think the idea is, you're probably not the first person to have thought of it. And the people who could have done it, either did or decided they didn't want to. And so you're running a relatively low risk in that respect. And so I think it's definitely worthwhile to do that. Take all the input you can, especially when it's free.
yeah, definitely. So maybe I'll pass it on to Rob. He has a couple of
Yeah, thanks, Nathan. Matt, always a pleasure to have you on. Fun anecdotes and a lot of insight. It was great to listen in. Also, you were on with your brother back in February, had a sort of an impromptu conversation where you talked about a lot of this stuff here. So I was thinking to follow up a little bit on just a couple of points. One is, you're talking about how just now you were saying you were approaching, like you have all these ideas and you talk about them with everybody, and generally they'd say that you're crazy, won't work, etc. Then you go to other experts who would try to resolve these things. So that's a very good strategy. But say you're someone like one of the audience here, or like us, is thinking about doing something similar to what you've done. How would you start? Like, could you just spend a couple of minutes to walk us through like the actual steps of approaching a researcher, an expert, how much time it took you, how many rejections you got, um, how long to write your emails, how much homework to do, that kind of stuff. Because I mean, it's not obvious at all how you would go about doing what you did.
Yeah, and well, I mean, every journey is going to be different, and certainly the journey you take, for example, if you have a piece of technology coming out of a super prominent lab, is going to be different than if you have no technology or technology coming out of a kind of obscure lab, because people will give you different, they'll give you the benefit of the doubt maybe on one thing, or they'll be more willing to take a call and that kind of thing. But I mean, what was interesting, I guess, is that when I first started this, the general attitude was that I had no business being in there. I didn't get like a PhD in molecular and cell biology or genetics or anything like that. And I'm here trying to wage war on something that people have been trying to do for a long time and failing at. But at some point in that journey, I crossed this like invisible line where suddenly I wasn't an outsider anymore. People knew who I was and they had seen some of the stuff we built, and they're like, "Oh, Matt, he has this cool lab and does all this fun stuff. And I had my own lab. I became like a rogue PI of sorts. I wasn't really beholden to any university or the government for funding, and so I could do all sorts of fun things that they couldn't. And so I say that because how the world looks at you is going to impact a lot what you need to do to get started. So if you're already like, you've done a couple of postdocs in this, you've written some papers, you got a couple high impact journals under your belt, like you're going to take a different path than someone like me who didn't have those things. But I'd say that assuming take that difference away for a moment. What I normally do if I'm contemplating a new project is, I have a ton of these ideas. In fact, I've been looking to hire somebody with the express purpose of helping me run down a bunch of these ideas without distracting my core scientific team. And so I frequently have something that sounds really cool. And then the real thought is to say, okay, well, who's done work on this in the past? Choose target X. And maybe at some point in the call, it might make sense to have someone who has an actual example we can kind of walk through because it can be a little easier that way. But I guess the first is map out what's been done. Like, who are the experts in this kind of field? That who are the ones who you might want to talk to, who are the ones you might want to avoid, for that matter. I mean, some of them are can be kind of assholes. So it's good to understand like who's in the field. And then you say, okay, well, if I get this technology and assume I can make it work, how would I apply it? Is there like a viable path to turn this into a drug that can make a lot of money, be sold to a pharma that can actually address a meaningful need? So you have to kind of look at these through two different lenses. One is like a scientific plan to validate your idea and move it through from being in a dish to being in an animal to having conversations with regulators to human trials. The other is answering the question of, even if I assume all that stuff goes well, what am I going to do with this thing? How is it going to make money? How is it going to help people? Can it be made at a scale that is viable, or is it likely to be viable in at least in a decade or so? And so I think these are the lenses you have to look at it from, and which one you spend the most hours, say, doing homework on is really going to depend on where you're have the most unknowns. Like if you know everything about this molecule because you've been working on it for years, for example, you're not going to have to do much of the background work on that. You probably already have a good idea of how you're going to need to run it through the paces to validate it in a more advanced model. Then you're going to need to spend a bunch of time on figuring out if I can make this, like, for kidney disease, right? To look a lot about, the kidney disease market, like how many people are there who have this? How many are, identified, and when are they identified? And how many, like in our case, we decided to not go after diabetic kidney disease initially. And that's actually the bulk of kidney disease patients. But and I think it would probably work in those patients too, really. But it added another layer of complexity to it that I didn't want to tackle in the early trials. I wanted to have less risk in my early clinical trials. And so we, talked to all the experts in that case, American Society of Nephrology, all these guys, all the key opinion leaders in there, and said, this is what we're trying to address. Like, where's the biggest pain point? Like, where should we get into this? And then, I posed this question a bunch. I said, what kind of data would I have to show you before you are the one advocating for me that this should be put in your patients? And it's like a blunt question that I think you should ask a lot of clinicians, and especially the kind of clinicians who do experimental medicine. But say, like, I've got compound X sitting here. Like, doctors are generally speaking fairly risk averse. They this whole Hippocratic oath thing, right? They don't want to kill their patients, and where scientists are in the habit of putting strange compounds into furry creatures all the time. And so if you frame it like this is the data I've got today. Like, what would you need to see before you would be advocating for me? And something that happened really interesting in my first startup in particular was that a lot of the people who were like the harshest critics early on, who were just like thought it was dumb, like they would invest a surprising amount of time telling me why it was so dumb. But some of these guys actually became some of the most vocal advocates for it of anyone, because they kind of watched this progression as like systematically the objections like fell down basically, or we found a way around them, and then they're like, this is actually pretty good. And then they become your advocates. And I think that's a perspective that you're going to want to take, but you have to, I guess the first step is looking at yourself and your goals and figuring out where the holes are. I guess that's if you were to abstract it to one thing, because the details about how much time to spend or who to talk to or what to do are all going to be based on that.
Thanks. Yeah, I mean, a detailed case study obviously would be very helpful, but let's just try to make this as practical as possible. Let's say for a potential grad student or a postdoc or someone, when you got started, where you said you came from a CS and physics background.
Mhm.
Were you self-funded? Were you writing on savings? Did you, like, from a
Yeah,
practical standpoint, how did it take you like 2 years before someone finally said, okay, this is interesting. You've talked to all these experts. Now we can fund you. Like, how
yeah,
long
yeah.
did
So
it take?
yeah, from my perspective, like I said, the idea was I wanted to build an app store for the human body. And actually, I'll walk you through like use as an example. So the thing that got me interested in biology initially was vaccines, and that's if you got a vaccine, suddenly a disease that used to be able to kill you wouldn't even give you the sniffles. Like you could take a lethal disease, and with this vaccine, if you got challenged with the disease, nothing happens. And I thought that was pretty amazing. Like, I mean, nothing's fundamentally changed about the pathogen or the host. Like it, but suddenly lethal goes to nothing. And everyone would, I guess, pay lip service to this idea of immune memory, which seems to have been forgotten, I guess, in the days of COVID. But that your immune system has a memory, and if it if it sees something once, it can fight it off. But I got asked, asking kind of as a computer scientist, is well, where's the file? Where's the hard drive? In what language is this written? Because I'm pretty sure immune memory, doesn't live in your brain, and your genome in general seems to be the same. So like, where's this? And it turns out, it actually is encoded in DNA, like in the variable and hypervariable regions of B cell receptors and T cell receptors in a subset of cells. But what my interest was in this initially was that what if we could take leverage the computational power of the planet against a virus. So if you think about how your immune system works in that respect, it's kind of a brute force attack. You randomize your immune cell repertoire, you make sure it doesn't attack you, and then you turn it loose, and then you hope it sticks to something. And when it does, it begins to mutate these things even faster in an effort to like build a better bind onto it, to have higher affinity binding. And so with certain diseases like HIV that we haven't been able to vaccinate against, certain people still manage to actually become immune to it effectively, or to produce antibodies that kept them safe, or they would get sick maybe very slowly or sometimes not at all, but super rare. Like, we've lost what 40, 50 million people to HIV. Like each of them made countless trillions of these antibodies, and only a handful got the upper hand. There's a brute force attack with flesh and blood. And so what if we could port these algorithms into cryptographic hardware, like these really powerful password crackers that people like to build? And that's actually how I got started on this, is trying to port Rosetta Doc to cryptographic hardware, to FPGAs in that case. And as I went down the road, one of the things that someone pointed out was that cells don't have USB ports. Like, you can't just upload it. Like, we already know what antibodies protect people from HIV, but there's no way to upload it to the person. And this is just, I suppose, speaking to my general ignorance of the field. Like, I had assumed that seems so obvious that someone would have done it before. It's like, how can you not have a way of programming cells? And so then basically said, okay, the focus is going to be how to program the cells. Like, forget docking proteins and all that stuff. Like, someone already has a sequence we can use. We need a way to upload it. And then it became a search for what tools exist to modify cells. And so that was like occupied probably a good year of research trying to like analyze the technologies for like getting information into cells. And eventually I kind of settled on these two technologies that I thought I needed. One was a measles pseudotype lentivirus that's found in France, and the other was this culture system for growing plasma cells from Caltech, David Baltimore's lab. And so basically I got licensed to that, or an option to it, and then put together this prototype virus, and away we went. And so early on though, I was basically just maxing out credit cards and stuff. So I had some money from my previous startups, but I was just trying to like buy as few things as I could, obviously, um, but yeah, just all my own money. And it wasn't until we basically had gotten these first little, glowing cells in a dish, GFP positive, primary B lymphocytes, that we got initial funding. And in my case, we had a couple early angels came in, and who were medical doctors, and then Breakout Labs was our first primary funder. So this is Peter Thiel's outfit, and we were actually the first in the first cohort of Breakout Labs, and they had this really interesting kind of mission. It was basically to fund things that were too too outside the box for the NIH and too early for venture capital. And so when we saw, like, they'd actually had a press release when they got started, and one of my guys saw this, and we looked at it like, if we're not going to get funded by that, we're not getting funded by anything. This seems like it was written for us. And so we applied, and that gave us first funding, and then we got an NIH grant after that. And basically, it allowed us to, I think our first round was worked out to be a little over $1 million, and we used that to generate our first data in mice, and then we raised more money and went from there.
Okay, so if I understand, excuse me, it sounds like you had some prior, like earlier startups that were outside of biotech, and then in the beginning you were sort of feeling your way around. Took like a year or two at least, where you were sort of self-funded, and then with enough persistence, it sounds like you eventually got through this Breakout Labs funding, and then the rest is history. Is that about right?
Yeah, that's about right. And I mean, for me, I guess distinct disadvantage in that it took a while just to be able to read medical journals fluently, right? Like when I first picked up, big article in Nature or something, I had to read these things with Google open in the next window to look up all the words. And so I didn't have like the background and the nomenclature and all this stuff. And so there was a little lag time just to even do that well. And then I got proficient at mining medical literature and had to make friends with all sorts of people who were at academic labs to send me lots of papers. And it's before the world of Sci-Hub and stuff. And that I'd say actually back then, my general position was that, like the paywalls on medical journals were the single largest obstacle to the advancement of science in the world. I mean, I think it's still a complete abomination, incidentally, that, taxpayers pay for all this research. Like NIH is the largest funder of biotech research by a considerable margin, and we can't even read the results of the data we paid for as the average taxpayer. It's complete bullshit. But yeah, back then it was I think that was even before Gigapedia, which like predated Sci-Hub. But yeah, so it was actually I had considerable delays just getting papers, because I'd read a bunch of papers, find a bunch more papers I wanted, have to like email all my long-suffering friends to like collect more papers and then go do it again. Or I would sometimes drive to my old university, and I would sit outside the library because it'd be closed at that point, usually in the middle of the night, and I would just slurp down all the papers from the Wi-Fi and then go back and read them all. So I make these big lists of all these things to get. But yeah, so I think there is definitely a lead time in this, unless, of course, like, say if you're already sitting there, you're quite familiar with what you have and what you want to do with it, a lot of that stuff would go away. But even in my position now, like when I get, new things come across my desk, usually I still have to dig into understanding the background of it if it's not something that I already was quite familiar with.
All right, great. So we're running short on time for the first hour here, so I guess pass it back to Nathan, or we can open it up to the audience here if there's anyone that has any questions.
Yeah, so right before we get into audience questions, so anybody in the audience, you can start raising your hands. But Matt, is there anything that we can do for you? Are you guys hiring, or you're raising money or something like that? I'll just let you put out a call for that.
Oh, well, I'd say in general, we're always looking for money, brains, and molecules. It's my mission. And so right now, like, so we're closing one round. We're going to open another. But we're always looking for new people who want to support what we're up to. I expect we'll probably be hiring more scientists relatively soon in our San Diego lab, because we just got a much larger space and it could be able to fit more in. But we don't have like any specific listings out. But I'm generally anyone who's interested in the space or has kind of cool approaches to it. Like I'm always looking for things to do. Like we've built this hammer, the ability to upload kind of arbitrary nucleic acids, and we have broad rights in the field of aging, and so in oncology. And so if there's things that can be applied with that can benefit from, systemic delivery of nucleic acids in particular, I think that's we're always interested in talking about that.
Okay, great. Yeah, and if you have any job listings, just email me, and I can post them on my website as well. Okay,
Awesome.
cool. Let's get to some questions because we went way over. I mean, Matt, you had awesome answers and definitely a gold mine for advice for people who are looking to get into this. But let's start with the first question. Attila, maybe you can just introduce yourself and then you can ask your
Sure.
question.
Sure. Thanks a lot, Nathan, and thanks a lot, Matt, for speaking. My name is Attila. I work at Blue Rock Therapeutics in Toronto. I'm a research associate. Yeah, so basically, I just have kind of like maybe a broad question. Given your experience and expertise, where do you see the longevity industry go? Long term, like 20 to 30 years. And do you think that we can reach like the longevity escape velocity in that time? Thank you.
I think in general, like the future is in synthetic biology. Like we're having to do more and more complicated things and control things that need much more precision. So I think especially as you begin to look at things like cellular reprogramming or rejuvenation of tissue in situ, I think that becomes much more critical. The other that I think moves into I guess you have two lenses of the longevity thing. One is get rid of the damage that causes problems, and the other is like actually drive rejuvenation. And Senolytics being an obvious first start on one, but you have all sorts of things like cross-link breakers, trying to correct metabolic dysfunction and things like this before you kind of get to actually driving rejuvenation. And so, but I think synthetic biology is going to become an increasingly important role in this. And I think that there's a sporting chance. I mean, we've roughly doubled our lifespan in the last 100 years or so, plus or minus. And I don't think the gains will be as easy going forward. But I think if you look at just the Senolytics on average extend lifespan in animals by around 20%. I think that's a ballpark that we can probably get. Maybe we get half of that, but in a human. But I think we can definitely make progress. And with every piece, my attitude was they'll buy me 10 more years to think of the next problem.
Great. Okay, so let's go to our next audience. Oh yeah, and just FYI, anybody coming up to the stage to ask a question, it means you consent to us using your audio in the recording. Okay, so everything is being recorded. King Yin Yan.
Hi, I'm an independent AI researcher, and I have been working on a theory of AGI for some many years, and I want to join a project to apply my AI. And also, I have some background in biotech. So I would like to know what's a promising area to apply AI in longevity research.
Well, I think this is a bit out of my speed. I'd say actually, the BioAge crew and in Silico crew are probably the people to ask about this. They know better than I do. But I mean, on one hand, I think they're mining things like both biomarkers of age and changes of things that you can detect from biosamples, and on the other, basically trying to anticipate things that would likely be therapeutic. And these, I think, both have applications. One thing in my mind that I think has a lot of promise is actually more basic, and that's just better ways of mining medical research. And I mean, there's so many papers written that at this point it becomes impossible to keep up with them at all. And the ability to mine larger and larger data sets for like natural language processing, I think, has a lot of benefits. But yeah, I would probably hit up Alex or one of these guys. They're the ones who would know this the best.
Thank you. And also, I would like to mention something like AlphaFold, which is like a small problem that has been solved with AI entirely. So, and maybe something more like nanomedicine. I mean, like designing the molecules, which is
Yeah.
seems to be suitable for AI.
Yeah, I think that's probably something more that Alex is doing than Kristen, but I'm actually not certain. Due to all the lockdown, I have not been talking to them recently. But those are the people who come to mind the first in the field that I think are worth talking to.
Okay,
Yeah.
thank you.
Yeah. Okay, great. Yeah, so you can definitely check out Alex Zavarenkov's company, In Silico Medicine. Okay, so next question, Joshua.
Oh, hi there. Joshua Youngkin. I'm an attorney in San Diego, and I worked with Matt back in the Discovery Institute days a while ago. How are you doing, Matt?
Doing great. How are you?
Doing well, bud. Hey, so I didn't catch the entirety of the conversation, so apologize if this is, if you already covered this. But did I hear you say recently that you guys plan to come to San Diego?
Yeah, well, we actually have a lab in San Diego, or technically Oncocenix, our oncology spin-out. So I'm actually in Torrey Pines right now. But
Oh, wow. Okay.
so
Yeah.
we're we are at J Labs initially, and we still have a small J Labs footprint, and then we got our own lab across the street basically, and now we're moving into a much bigger lab across the street from that.
Oh, wow. That's incredibly exciting, man. How goes the patent profile? Are you guys constantly filing? What are some of the challenges there?
Yeah, it's I think the patents have like a special kind of frustration where you patent something and they're like, oh, that's impossible. Then it switches to being obvious. And there's it is yes, it drives me insane. But we've managed to get some early patents issued already. The delivery technology has patents that go back from before I got my paws on it, and then a whole bunch of new ones on the updated variants of it. Like the delivery technology improved by like 80-fold in the time we've been dealing with it. So it's pretty much a continual barrage, and a lot these days on kind of applications and new payloads and that kind of thing. But yeah, I've spent an embarrassing amount on IP attorneys. I'll tell you that much. But I should have a boat with my name on it somewhere.
Well, that's actually my last question is what's your exit strategy?
On the oncology side, it seems pretty well set up to license or sell to a big pharma company. It's really straight down the center of the fairway for that. And so a lot of even the IP strategy has been built with that in mind. On the aging side of it, we're much more interested in building, a Pfizer for aging than we are selling it. And so we want to, so we have applications that go well beyond analytics, like a very broad mandate to all sorts of things, some of which are right now would seem pretty far off, but I think, in time will become much more accepted. And so goal is to like build things like we might, for example, license out the kidney disease application, but we're going to keep pushing the platform forward, ideally in-house on that side.
Very interesting. Do you have any specific buyers in mind?
Yes, although I can't talk about them.
Okay. Sorry. Okay. Maybe another time.
Yeah.
Okay, great. We're at 5 o'clock now. Matt, do you have time maybe just for a couple more questions?
Yeah, I
want to
can
be
stick around
listening.
for a bit longer.
Oh, okay, great. Thank you. Okay, so John, what's your question?
Great, thank you. Hi, all. This has been really helpful, so thanks a lot, Matt, for doing this. But my name is John Young. I'm an MD, PhD, looking to transition into the longevity space, and really interested in gene therapy. So this has been great. But my question is, what innovations do you think would be the most helpful for further improving gene therapy for aging? You mentioned kind of the delivery vehicle, and you spent a lot talking about that earlier in the discussion. And is that what you think where more innovation is needed, or our understanding of aging, or maybe even finding better targets or multiple targets?
Yeah, well, delivery seemed like it was like the harshest, the most acute pain point, really, and it's why I put so much effort in. The last decade of my life has been obsessing about how to get DNA into cells, and I'm quite optimistic about our particular delivery technology, and I think it's going to open up an awful lot of possibilities that weren't really there before. Because if you can't master delivery, having the the perfect editing tool isn't very useful. Like if you're only limited to what you do ex vivo or like hitting the liver repeatedly, you definitely have, I think, problems with that. But so, and I think that even from our delivery point of view, it's definitely a moving target. We're working on, always evolving this thing to, be able to hit more cells more reliably. And on the payload front, I think some of these tools like, durable episomal vectors, like I'm a big fan of the nanoplasmid. Some of these things I think open up the possibility to do things that you couldn't really do before without the risk of having to do insertion. And so some of these basic building blocks are coming a long way. I do think that in terms of aging, still the biomarkers aspect of it, like how do you measure success, is still a big problem. I mean, that's why we're pretty much stuck with going after targets that the FDA likes at the moment. And I do think they'll crack at some point in terms of treating aging itself as a disease, or at least a target you can go after. But for now, it's we go after kidney disease or idiopathic pulmonary fibrosis or cancer, and because you can see the result basically, and so being able to understand the mechanisms of aging and how to see its progression and ideally its reversion are really important tools, and that we're beginning to get at. I mean, the biological clocks, I think, are kind of the epigenetic clocks are these interesting, somewhat emerging class of biomarkers for it. But I think the way they're trained, of course, biases them one way or another. And none of these things have a proper reference standard in the sense of like, we've never shown that we can take an old person and make them younger. So how do you know if it's working? You can, they do seem pretty good at telling you if you're in bad shape or relatively in good shape based on your age, but not so much measuring the outcome of an intervention. So, I mean, a Senolytic's a good example. There are definitely some of those clocks that respond to senescent cells a lot, but others ignore them in their entirety, basically. And part of it would have been on the cell you sampled anyway. But so being able to get a better handle, and probably given your expertise on both the medical and the science side of looking like how do you address this from a patient's point of view, like on for so much of history, aging medicine has been kind of the purview of snake oil and cosmetics, and that you were kind of prohibited by definition from making a cosmetic that worked because it would become a drug, and if it's a drug, it would have to be regulated and then have to have a disease. And so it's kind of interesting to look at how companies have skirted that over the years. Think of like Botox, probably the classic example of this. Implant restless leg and moved into wrinkles. But yeah, I think being able to find good ways to actually determine what's progression and what's not, probably for the field has the most value. In terms of actual new therapies, like I'm actually really interested in the things like the cross-link breakers. I think they're largely an orphaned part of the space. There are a couple of people working on them now, but this idea of how do you deal with extracellular junk, like all the stuff we're dealing with kills cells that are defective. They don't deal with remodeling the extracellular matrix or tissues in general. And so one thing that we're looking at ourselves in the future is dealing with that, and even things like collagen deposition, and that is admittedly a little more cosmetic, but still is these things of how do you actually age? What's the difference?
Yeah, great. Thank you.
Okay, great. Next question, Eduardo. Oh, okay. Maybe we'll go to Anar first. Anar.
Oh, hey guys, can you guys hear me?
Yep.
Oh, and hey Matt, really interesting. By the way, I'm a CEO of Ageless RX, and it's really interesting to hear all the hustle that went into building your company. I can definitely relate to that and appreciate it. That's amazing. Question in regards to the mice, right? So it's interesting. So this is the senescence treatment where the mice are already aged, and even the aged mice end up living longer, right? Like you don't have to start young because you don't have senescence burden when you're young.
Yeah. So I mean, one of the interesting things about our version of the study was that we started in mice that were quite old. They were 2 years of age, and so this is like equivalent to like a 70-year-old person at this point. whereas the Mayo one, I think, started at it's 18 months, I think was their old cohort, if memory serves. It is younger though. And there is an interesting thing. There's certain kind of pathologies that you can prevent, but you can't undo. I mean, an interesting one from theirs when they were treating a young age is they could prevent a cataract, but you couldn't get rid of a cataract later. So certain tissues can regenerate better than others. And I think, the general thesis behind senolytics is that the senescent cells are actually impairing your ability to regenerate, and taking them away just allows your body to regenerate better in general. I mean, something that anyone could observe that like kids tend to bounce back faster than adults. A kid breaks a bone, hurts themselves, they heal faster. They can tolerate much more damage and rebound from it than older people do. And so the thought is that this is taking a breakaway of sorts, not necessarily improving regenerative capability beyond what it was. And although I would say that if you were going to choose an optimal time to start taking senolytics, it would be more on the age of like 30 from a human than it would be at 50 or 60. And because, that's kind of the rough time when you start, stop growing and start dying, like it's kind of interesting, right? A human can go for decades without getting a wrinkle. And this has always kind of captured my imagination that you have this ability to tolerate a fair amount of abuse. I think you do accumulate senescent cells early on in life, but they pick up pace exponentially as you age. And if you look at your genome at 80, it's basically the same as it was at 8. And of course, there's somatic mosaicisms and you accumulate some mutations, but on average, you have still plenty of cells that have more or less your core genome. But you're in a lot worse shape at 80 than you are at 8. And I mean, the reason you got that wrinkle wasn't because you forgot how to make the collagen. It's that something was actively blocking the process. And so that's, I think, why I think actually targeting younger on it might be beneficial, even if you're not seeing symptoms.
That's really interesting. So it might be interesting to see if you start targeting younger mice, if the survival improves even beyond the ones where you start older mice.
Oh yeah, I mean our reason for going old was a little bit pragmatic, and it was that mostly rich people tend to be old, and it wasn't like the FDA is going to let you put an aging treatment into a toddler anyway, so you can't go after a kid that was just weaned and give him this. And so we selected this out of, out of kind of pragmatic reasons that weren't strictly academic.
That's interesting. Got it. And then just one other question is, how often would you need to administer? Like in mice, is just one time IV or injection, and also even eventually when these mice die, they die from the same reasons as the control group, or are the reasons different?
So we were injecting them once a month. In humans, I would think that if you started the treatment when you're old, you might want to take a few of them in relatively short succession to try to knock down this larger burden you have. But then you're probably only going to need to take it every few years, really, if I were to guess. Like, assuming you have a good senolytic that can actually take out the majority of the cells. The on the what's the second part of that?
Um, they die for different
Oh,
reasons,
was it
right?
dive? Yeah, yeah. So for the most part, yes. They would. You'd see some of them would get really aggressive cancers, and some of them would just keel over with no real determinate cause. But you'd see things that looked like liver disease and cancer in some of them, which is something you'd see in the controls and the old mice. But it didn't seem to skew it really hard one way or the other. So they were. In the end, it didn't make them immortal, but they didn't always have like really obvious causes of death. I mean, in our study, like I think 3 of them were sacked for atopic dermatitis, basically, like which is ridiculous. You don't usually sack your grandma if she gets a rash, but we didn't have treatment for it on the protocol. So if they got a rash and it got bad and didn't resolve, they sacked them. But
Got it. Awesome.
so not super helpful. We had a much larger study in that we started looking at next, and that was basically trying to look more at things like cognitive metrics and like health and frailty. So basically what kills a lab mouse in the end didn't seem to change a lot between them. It just happened later and more abruptly. But I don't know how much it's worth trying to go after those things in the sense that they are a lab mouse at that point.
That's awesome. Got it. Thank you. Thanks so much.
Okay, thanks for your question, Anar. I think we're 10 minutes past now, so wait, oh, we have one question. Eduardo, he's back.
Hi.
Okay, last question.
Yes, my question is, I don't know if you know a recent study of the Back Institute where they have showed a big relation between the senescence and the Alzheimer. And I don't know if all of this technology may be applied to the Alzheimer, as there is a strong relation according to these studies.
Yeah, so I'm not sure about Alzheimer's proper, but we were showing improvement in cognitive function in the animals that we were treating, and so and we're digging into this more on like an academic basis, trying to understand a mechanism behind it. I mean, Alzheimer's is a more, of course, specific form of dementia, and we weren't looking for the Alzheimer's models in mice are different, so these are just normal mice, but they did seem to have improvements when you treated them with this, and so I think there's likely something there, although I don't understand it academically.
Okay, maybe we'll just wrap it up with one last question. Thank you so much for your time. But Carmen, if you can just make one quick question and then.
Yes, thank you very much, Matt. That was very informative. I just wondered, you mentioned your experiences with interacting with other scientists over time, and you alluded in some ways to experiences with regulators or interactions with regulators. But I was wondering if you had any observations on the kind of evolution of the regulatory approach to what you're doing or what you might be doing over the years that you've been involved in it.
Yeah, so I think probably the biggest one from a practical point of view for me is the acceptance of gene therapy and genetic medicines in general. I mean, this has changed a lot, but I mean, I'd say actually probably the biggest change that no one has really, I think, wrapped their heads around yet was what just happened with the COVID vaccine. I mean, if you think of like the AstraZeneca, J&J, these types of vaccines, like they're a straight up gene therapy. I mean, it's a live virus delivering DNA. And the thought that you would basically be giving gene therapies to healthy people a few years ago, I think would have been, absolutely out of the question. Like no one would have tolerated this. And I mean, gene therapy has been reserved mostly for terminal patients and rare genetic diseases, and it's been moving slowly into things like hemophilia and stuff that aren't necessarily going to kill you right away, but certainly not being done to healthy people. And I think that this is actually a pretty significant shift. And my hope is, of course, that it will go well and it will make open up the regime a little bit more to genetic medicines. But and so that kind of progression. I mean, when we had started Immusoft, the first company, there was no approved cell therapies at all. And there's no CAR Ts. This is before even Provenge, Dendron. And so in that time now, there's I think something like 900 gene therapies working their way through clinical trials right now. It's huge, and so that's a big deal. On the aging front specifically, I think like the TAME trial and stuff are an interesting development from a regulatory point of view, in that it's one of these. It's an early instance of the regulators accepting kind of a constellation of endpoints that I think this kind of endpoint would be necessary for aging if you wanted to get an approved drug. And so I think it's making progress towards that. But yeah, I think that it'll come down, I suppose, to demand. I mean, the FDA is fundamentally pretty risk averse, but it's balancing not wanting to have patients upset because they can't get a drug, and not wanting to have patients upset because they got killed by a drug. And those two kind of factors, I think, drive a great deal of what they do. But the much greater experience we're having with these advanced kinds of treatments, I think, is going to make life a lot easier in terms of having a predictable path that everyone can follow.
Thank you. That's helpful. That's very helpful. Thank you.
Great. Okay, so thank you again, Matt, for coming on the show and answering all these questions. I love the talk that we just had. It was really informative and lots of practical advice for potential founders who want to get into this space. Just a reminder, anybody out there who's maybe a research scientist and they're interested in getting into this field, Matt mentioned that they will be hiring at some point. And of course, more money is always needed. So if anybody is an investor, or you know someone who's an investor who might be interested in Oncocenex or Oisín Biotechnologies, definitely try and reach out to Matt. So without any further ado, I think that's basically it. We'll close up here, and then maybe we'll open up a different room just for some casual after chat, just for maybe half an hour. But yeah, so once again, thank you, Matt, and I think that's what we've got.
Yeah, thank you, Nathan. Robert, it's a great time. Thanks for having me.
Yeah, definitely.
Thanks.
Okay, let's wrap it up.